uh, getting into this Google Meet. So for all the participants who are here, for you that before introducing Nupur sir, just a brief introduction about the Science Dialogue series. It's a brainchild of Dr. Gopal sir. It is initiated approximately 6 months back, and with the concept of making the knowledge available to everyone and each of us who are belonging to the science, and we are contributing a bit of ours to the science. This lecture series, it is actually taking care of the spreading of knowledge from the eminent scientists, from the eminent personalities of the science, who are there to contribute their knowledge, their experience to the students, as well as to all those who are interested and willing to join this science dialogue series. So, with this brief introduction, I would like to introduce you all with the today's eminent speaker, uh, Dr. Nupur Das sir. So, for all of us, I can say that this is the first time when actual doctor from the MBBS background, who has also pursued his what should I say? He has also pursued his PhD or doctoral degree in this field also from the JNU School of Molecular Medicine in year 2000. So then initially he had done his MBBS from the Chittagong Medical College, University of Chittagong. Then he carried out his PhD in the Molecular Medicine Special Center for the Molecular Medicine, JNU, under the guidance of Dr. Chinmay Mukhopadhyay. Actually, from starting, his interest was is related to the iron metabolism, where he has carried out his PhD with a thesis title "Studies on the Regulation of Iron Uptake Gene Transferrin Receptor 1 in Host Macrophages During the Infection by the Parasite Leishmania." Then moving ahead, he did a lot of postdoctoral training. Just after his medical MBBS, he has worked as an internal medical officer. Then he has done some of the experienced residential medical officer in the Bangladesh. Then moving ahead after his PhD, he has moved to Ohio in Cleveland, postdoctoral research fellow at the Department of Cellular and Molecular Medicine. Then he carried out his another postdoc at the Department of Molecular and Integrative Physiology, University of Michigan, Ann Arbor, Michigan, where he is currently working as a assistant research scientist, professor in the field of this molecular and integrative physiology, University of Michigan. So his whole career is revolving around the study of the iron metabolism in one or other way. He has published a number of articles, research articles in very prestigious journals. His citation speaks a lot about his research caliber, with more than 1,500 citations to his caliber. He is a regular membership. He's working as a regular journal reviewer board on the very reputed journals of the nutrients and metabolites. Apart from that, he is also providing his peer review journal services to the Royal Society of Chemistry, then scientific reports, Journal of Biochemistry, JBC, food and nutrition, nutrients, biochemical journal, and a lot of more. More than approximately 15 journals are there, international reputed journals are there, to which he is contributing as a peer reviewer. And what to say more, he has published a lot number of research articles in the same field of the iron metabolism. So I think that all of us, including me, as well as all the other participants who are present in this science dialogue lecture series, they will be definitely benefited from his research experience, vast exposure to the field of this iron disorders, how they can be treated with the novel strategies. So, with this introduction to the our speaker of today's session, Nupur Das sir, I'll be handing over this virtual platform to sir, so that we can start the formal session with his lecture. So, sir, now this platform is yours, and our participants are also eager to listen from you that how we can look forward for the novel strategies to treat the iron disorders.
Can you hear me all?
Yeah, hopefully everyone
Okay.
is able to listen you.
Great. Thank you so much for this beautiful introduction. I'm feeling very nervous now that I don't think, I have achieved that much. I'm still trying. But what I would like to say is that as a physician, I've been a physician in Bangladesh, and from there I've traveled. I changed my career, and all through the career, I would say that I just got stuck with iron metabolism. I just cannot get out of it. So I'll try to explain why it is important to target these disorders, why it is still challenging to target these disorders. I'll try to tell a story, and thank you in advance for your attention and for your time. uh, if you feel like to interrupt me, please, if you have a question, I'll try to answer that. I'll try to keep it as brief as possible, to in a way explain things. But if anybody needs further explanation, please, interrupt me, and then I'll try to answer as much as possible. Having said that, before I start the presentation, go to the presentation mode, I'd like to turn off the video. That will give me, uh, some much comfort, a little bit more comfort to handle the slides. So
Yes,
thank you
sure.
for that. Yep. Uh, trying to copy. One second. For some reason, the okay, got it. Turning off. Second. So can you now see the presentation mode?
Yes, it
Thank you.
is.
Yep. So the title of this talk is the novel strategies to treat iron disorders. The iron is required for nearly all living organisms. The fun fact is our body. One second, let me just start the laser here. Pointer. So our body has enough iron, to make a metal nail about 3 inches long. It sounds like a very little amount of iron, but that is, essential to make RBCs or red blood cells in our body. 70 to 80% of iron is required to make RBCs. And as you all know, I don't have to emphasize in this group, but hemoglobin is the molecule that needs iron to make it so that it carries oxygen to different parts of the body. I will come back to this later, but just to emphasize, the iron and oxygen are very intimately connected, and that's why iron metabolism and oxygen metabolism are intertwined with the same, pathway. The rest of the iron, although if you look at the percentages, it doesn't build up to 100%, it's more than 100% because they overlap. There are a lot of overlapping between these percentages. So but rest of the iron actually take parts in the metal, so more than 30% of the protein are metal binding protein, and metals are involved in 40%. And you can say that iron is one of the most essential metal in our body. And not surprisingly, the iron, the plants, bacterial kingdom, and humans, basically except a few bacteria, iron is required for all living organisms. So the in our body, iron has to come from diet, and we do not synthesize it. So if you don't take enough iron through our diet, we'll have some health issues. And that's why iron imbalance is a problem, is a global health concern. As you can see in this balance, if you have a deficiency, you will not produce enough red blood cells, and eventually you'll be anemic. But On the other hand, if we absorb, I'll get back to that, what happens or how it happens, but eventually if you absorb excessive amount of iron, it will develop iron overload in our body. So let's just focus on anemia first. So what happens when you have anemia? These are the some basic symptoms of anemia. Anemia is essentially the low hemoglobin, and iron is needed to make hemoglobin. That's why we are looking at the different symptoms that is resulting from this in low hemoglobin in the system. But the main treatment of deficiency, iron deficiency, is iron supplementation, either by oral, iron tablets, or if in case of severe anemia, you can actually intravenously give iron solution so that they can be corrected. The This is the global distribution of anemia. More than 1 billion people are affected worldwide. And as you can see, this is the place actually where we belong to. So India, Bangladesh, and the surrounding part, and the most part of Africa are affected by this. So mostly the anemia that we see, of all the anemias, majority of the anemias are nutritional. So if you don't have enough iron in diet, eventually humans will be anemic. But there are some problems. More than 20% of the anemia that we see, iron deficiency anemia, are actually refractory. So no matter how much iron you take orally, the anemia still persists. So there are some genetic components to it. And the other problem in this part of the world is that it also coexists with infection and the anemia of chronic diseases. So what happens is and that's the essence of today's talk. What happens is, in this population, they also have coexisting infections due to, for example, tuberculosis. And if you give uh, to correct anemia, if you give drastic or dramatic level of oral iron, actually the infection will flare up, because in that case, those infectious agents will also take up iron from our system, and they will actually start multiplying. That's a problem. So these are the problems we have currently. The the other picture, when you're looking at iron overload, what happens is that the this is a healthy liver. So when our body is taking a lot of iron through the diet, and it develops iron overload, and you see it looks, these iron deposits are in patches and actually destroys the organ, because excessive iron is toxic. It produces reactive oxygen species and damages DNA and all the other molecules in our body. uh, the If you look at these hands over here, you can see this brown coloration is because of the deposition of iron all over the body. Inside it's like that, it is destroying the organs, and exteriorly from outside it looks like that. And the conditions are called hemochromatosis, means the coloration of the body coming from iron deposit. So again, the treatment is very simple. I would say still primitive. It's the iron chelation. Or basically you give a chemical which can chelate iron, and so take iron out of the solution. And the another approach is you do repeated phlebotomy. Phlebotomy means you do bleeding, and the patient comes to the clinic in 3 to 4 months. You take the blood out. With the blood out, a lot of RBCs coming out, and so is the iron coming out. So in a way, to reduce the iron overload. But as you can tell, the iron chelation is a pharmacological treatment. It's not very popular because it also chelates the useful iron. Repeated phlebotomy, who wants to go to the clinic every 3 to 4 months? So till today, these are the basic uh, you know forms of treatment. And if you look at the map, this map actually represent the opposite side, reciprocally opposite side, that we have shown in the previous slide. That's the global prevalence of the iron overload. The iron overload develops from mutations in the HFE gene, and these mutations, C282Y is the most common. By just looking at the sheer number, one in every 200 to 500 people are affected in this country, and globally, this is pretty much the picture. So iron is, essential for our body, but then if you have less, that's bad. If you have more, that's bad. So you need a very tight balance to maintain it. And when the balance is lost, here the cause is genetic. Generally our body is equipped to regulate it properly so that we don't take enough iron. HFE is the regulator. So when the HFE is lost, no matter, even in with a normal iron intake or low iron intake, you cannot control it. Over time your body will start, depositing iron all over the body. Let's say it is getting deposited in the pancreas. So you know that pancreas produces, beta cells, insulin. So through beta cells is produced insulin. So it eventually, the patient may report come with the diabetes and with the skin coloration. So, and if they're coming from this background, we have to think that they may have a family history of hemochromatosis. So, having said that, let's get to the systemic iron metabolisms. As I told, it has to be very well balanced because you need enough iron from your diet to keep the hemoglobin level normal, but you also have to control it so that you don't take excessive amount of iron. So here what you see is that intestinal, a section of intestinal epithelial cells. So iron has to come from diet. So dietary iron is in ferric form or Fe3 plus form, and with the help of ferric reductase, it is reduced to ferrous form, and then it's transported into the intestinal epithelial cells by DMT1. The full name of the transporter is divalent metal transporter 1, but from now on we'll talk it the DMT1. And after getting inside, it is actually exported out of the system, out of the epithelium, intestinal epithelium, to the systemic circulation by ferroportin. So these three transporters that I talked about, ferric reductase, DMT1, and ferroportin, this will be part of the talk. I'll keep on mentioning these proteins. So after coming to the systemic circulation, iron is bound with the iron binding protein transferrin. So iron alone is toxic. So that's why the transferrin bound iron will go to the different tissues. Here, the major tissues that are part of this systemic iron metabolism are highlighted. For example, liver is the main organ for iron storage. And if you look at it, the bone marrow, of course, we know that this produces RBC. And here is the splenic macrophages, where they are responsible for recycling the old RBC. So we don't have to take all the iron that we need for the day. Our daily iron demand is 20 to 25 milligram, but if we keep on having 1 milligram per day, that's good. But if you don't take this very short 1 milligram of iron, if sorry, it's just somehow sorry. So it's just eventually the recycling will not be able to help, and then will be anemic. The This is the one part of the systemic, and how iron is coming in, how iron is being transported to different organs, but it is regulated at the systemic level by a hepatic hormone known as hepcidin, and that's the master regulator of iron metabolism. How? So what happens is it produces hepcidin, and hepcidin will go into systemic circulation. It will target different tissues, and those tissues are expressing ferroportin. I just mentioned previously, from the intestinal epithelium, it is through the ferroportin that iron has to come through the systemic circulation. So when hepcidin is there, it will actually continuously, in normal scenario, in a physiological scenario, hepcidin will secreted, this green that circles that you see, it will come, and it will keep on continuously degrade ferroportin by binding, and will lead to intercellular degradation. So what happens is that then it can control how much of iron will come out of the systemic circulation. But in conditions of low iron, when our body is not having enough iron from the diet, what will happen? Eventually, it will send a signal to the liver, and then it will not produce, or it will stop producing or lower the produce of hepcidin. In that scenario, it is doesn't have any control over the, this control is gone now. Now you see more ferroportin expression, and it will actually help to restore the iron level by exporting iron out into the systemic circulation. So this is an adaptive mechanism. When you don't have enough, iron in your diet, hepcidin will be low, and then it will correct. But then in some conditions, let's say there's a genetic mutation in hepcidin or ferroportin, overall the hepcidin ferroportin signaling, what will happen? Your control is completely gone, and then it will actually rise to iron overload in mammals. So, and this iron overload, it characterized by intestinal hyperabsorption, because now this is lost, and then through the intestine, a lot of iron will be going to the system. So in short, if you have low hepcidin, you have more iron absorption. So let's remember that for this talk, that this is the master regulator of iron absorption, and you need it to absorb. And if you don't have it, you don't have any control. You're just, accumulating tons of iron in the body. Let's look closely at the regulation of intestinal iron homeostasis. So I know that intestine is the port of iron entry. I just highlight my current mentor Yatriksha. He actually published a seminal publication in 2009. He actually identified this whole pathway that uh, HIFs, or hypoxia inducible factor. At the beginning of the talk, I was mentioning that iron and oxygen are very intimately connected, and both iron and oxygen metabolism are regulated by HIFs. I'm explaining shortly. But if you look at this, that again, iron is coming through the dietary iron is coming, passing from lumen to the plasma or systemic circulation through this pathway. But within the intestinal epithelial system, HIF2 alpha is sitting over there, or hypoxia inducible factor 2 alpha, and is actually controls, and it is the transcriptional, direct target of these three proteins that I talked about. Ferric reductase is another name, is D side B. So D side B, DMT1 sitting at the apical surface or the luminal surface, and ferroportin, as you may understand now, the ferroportin is sitting on the other side so that it can export iron to the systemic circulation. So how the HIFs are regulated? So HIFs are actually, regulated by PHD, or prolyl hydroxylase enzyme, in a both oxygen or iron, dependent manner. So what happens is that in normal scenario, HIFs, will be hydroxylated when enough oxygen or iron are there in the system, and eventually it will be led for VHL mediated degradation by ubiquitination pathway. But when there is no, when this oxygen or iron concentration is getting low in the system, HIFs are stabilized because this system is gone now, and now it is open to form heterodimer with its partner, and then eventually this transcription factor can go and induce the target genes. So basically, there's a mechanism, and in the intestine, these are the three transporters. So we look at these transporters as a readout of HIF2 activity. So I will change the gear a little bit, because that's the so far we talked about the why iron is important, why targeting iron disorder are still relevant, and how iron is getting in, and what's the basic mode of mechanism of this pathway. But in this portion, we are not just alone. We're not just taking passively some food nutrient, although I'm talking about just iron, but we are talking about, we are actually taking all the components of the diet, vitamin, all the other micronutrients, in addition to the macronutrients that usually take. But here on these aspects, what we have, I'm sorry. So I just wanted to also show that HIF2, when you have low iron, HIF2 stabilized, but we can also detect this by Western. So going back to the our friends that we have in the gut, we have trillions of organisms, not only bacteria but also bacteria, virus, and fungi. But for the sake of our talk, we'll concentrate on gut bacteria because they actually constitute more than 90% of gut microbiota. So before coming to iron, we know that gut microbiota will also need some of the same nutrients that we take every day. But this is for this group, right, or this is already known to the scientific community that the gut microbiota plays a lot of role, not just locally, not just at the intestine, but for various diseases, from obesity, diabetes, to ranging from depression, uh, Parkinson's, and whatnot, basically. And to this point, I actually think that actually we are actually living for them, because the ratio of our gene with them is, I think it's 1 to 20. So the amount of gene that we have, we're actually harboring within our gut more than 20 times the genetic material. So if you just think about, for this part of the talk, that they actually need iron for their survival, and this iron is coming in our diet. The question is, how uh, do they crosstalk, or do they ever crosstalk with the host iron homeostasis mechanism? How are they helping us, or are they just stealing our iron from us? So when we started this project, the scientific community actually did not know. So so we are fortunate that we had this open question. So what we did actually to address this, we took a very simple approach uh, to understand what's happening. So we took germ-free mice and specific pathogen-free mice, meaning they have normal pathogens, but then sorry, normal commensals in the gut, but they are devoid of some of these pathogens that we should not have in the normal mice. So that's why the term is specific pathogen-free, or SPF. So, but to remember, they have normal gut flora. uh, on the other hand, we had some germ-free mice, meaning they don't have any microbiota, they never had microbiota. So, and they're maintained in a very strict environment, whether they don't have any contact with the microbiota. So that's why they're germ-free. So these mice having normal, commensals, these mice having no uh, microbiota whatsoever. So we treated these mice with uh, different diets, different iron diets having 35 particles per million. Actually, we also treated them with even 10 times higher, which is 350 particles per million, which you call iron sufficient diet. So, and less than 5, you can go as low as 5 particles per million, which you are calling deficient. After putting them on 2 weeks, question is, why 2 weeks? We know that within 2 weeks these mice will be anemic, and that's good enough. If you don't give them enough iron in their diet, within 2 weeks these mice will have low hemoglobin. So we looked at hematology and gene expression to see what happens. So if you're looking at this, I'll first draw your attention to these blue bars over here. If you see now, within 2 weeks, these mice, iron deficient mice, SPF mice, are being anemic as they should be. And if you look at this RBC volume, these are the two most important markers of anemia, of iron deficiency anemia. So their hemoglobin will be low, their RBC size will be low, and that's why iron deficiency anemia is called, microcytic, because the size is low, and hypochromic because they have less coloration or less hemoglobin. So this is low, but surprisingly, we saw that germ-free mice were not becoming anemic within this 2 time point. You see here, they're not being anemic at all. Their hemoglobin level almost look normal. Their red blood size, don't change at all. So we were intrigued by it, and we're thinking, what's happening? One of the explanation is, oh, maybe because this bacteria sorry, these mice don't have any bacteria. I told that there's trillions of bacteria, so they they're just not there, so they're probably taking in more iron. So but the explanation was not that simple. So we had to look at the transporters, as I just mentioned, that the transporters, let's say DMT1 and the other two transporters, are the indicators of HIF2 activity because they are the direct target of HIF2. So if you look at this SPF group, and we have this now three iron diets, 350, 35, and iron deficient. As you can see, progressively, we don't see it over here, but here, there is also a change over here. But the expression of the intestinal iron transporters can go really, really high when you have low iron, because now this is an adaptive mechanism. Now it is upregulated because it wants to increase iron into the system. So this is how it works. And this happens through HIF2. So, and that's the thing that we see generally, that when you put them on low iron, DMT1 goes up. But what happens to the germ free? I mean, we are shocked to see it goes even higher in this, to almost 100-fold. That was unprecedented. That was not known in the field at all. We could not believe. And just to tell you that we got similar response, just for the sake of this talk to save time, I'm only showing DMT, but the other two transporters also show the same pattern. They actually more upregulated in the germ-free system. So what is happening? And the question is, do you need to put them on low iron to see this kind of response? What is happening here? So and because the bars is so high, you could not see clearly. So you look more closely. Then we see that even basally, for example, these are actually normal iron conditions. 350, we put 350 because in a normal mouse chow, normal mouse diet in our mouse facility, iron is about 300 ppm. So that's why we wanted to see what's happening in this scenario. But we don't need more than 35 ppm for mice to maintain regular iron balance. So you can see, if you look at closely, even in here, in the germ-free condition, these transporters are highly upregulated. So what is happening? So then we concluded that probably gut microbiota is needed to suppress the HIF2 activity in the intestinal HIF2 activity at the physiological level. So when you have that, it actually suppresses it. So now there are several questions. This is the germ-free mice. They never seen microbiota. Do they have any physiological problem? The immunological issues, that is that what is causing? It may not be just directly to just iron. It may be just that they are uh, inherently having some issues. That's why this is upregulated. So we did not know. So what we wanted to do is that we actually wanted to do microbial colonization in the germ-free mice. It's very simple. What it is that we take, homogenize the fecal pellet from these mice, from this normal mice, specific pathogen-free mice, and introduce them via oral gavage. And in a matter of actually a couple of days, 72 hours to 120 hours, they actually saw a good colonization. We confirmed that. And then you can actually bring down to the, and we are calling it conventionalization, basically. That's why the term is GF conv. So you can actually bring the response DMT1 and all the other transporters to the SPF level. So that was amazing. So by giving them microbiota, you can bring it down. The mice are good, they're healthy. And what happens if we you know use an microbiota depletion model? So in these mice, what we did, we used an antibiotic, broad spectrum antibiotic cocktail for 3 to 4 days in the drinking water. You can actually bring the DMT1 or the transporters level, I would say the HIF2 response or HIF2 activity level back up. I would just want to draw your attention to that the response is not exactly where, because it goes up to like maximum to like 7-fold or 6.5-fold, but it was very high in here, 15 to 20-fold, for example. What happens is that these mice, antibiotic cocktail is very temporary. It does not deplete all the microbiota, and that's why the response is not that robust. So to confirm that this is indeed happening through HIF2, so now we look at this immunofluorescence staining of HIF in the intestine, also looking by Western, just to show that this is a positive control. As I mentioned, that VHL is the one where the HIF2 will be degraded. So this VHL, these are intestinal knockout VHL mice in the intestine. So you see the HIF2 is robustly expressed over here. And the normal mice or SPF mice, the expression is totally gone. So if the microbiota is not there in that microbiota, germ-free mice, you see there is even higher expression of HIV, and you can bring it completely down to the basal level by conventionalizing the mice or giving the mice the microbiota. And here is the Western. You saw that you see that in the Western also it is up. So then you conclude that looks like there is an active mechanism. We are saying active because you introduce the microbiota, you can bring it down. Active mechanism by the camel cells to keep the HIF2 activity low. So the point is how the microbiota is doing that. Before going to the next slide, it is again a data slide, but I would like to emphasize that we actually ruled out a bunch of mechanisms, and we are very confused actually. And it is not, you know, uh, if you're giving a dead bacteria, the response is not that you need live bacteria. But then we found out that if you give a bacterial component, actually metabolite, the small molecules that bacteria produce in the gut, or the secondary metabolites they produce when they're eating our food. So we tested that. To do that, we actually isolated the duodenal and cecal content, duodenal cecal content, because the iron absorption is taking here. That's why we wanted to focus on the microbial population or the microbial components from here. And cecal content means it is almost feces or stool at here. So it has also a lot of microbiota. So we took both of them and then process them. This is a very standard process to extract, the crude metabolites. So methanol chloroform water extraction, uh, and then use those metabolites to treat the colon cancer cell lines, or they represent because they express the exporters. They have the HIF2 expression. So we did HIF2 Western assay in HCT116 cell lines. So on both sides, actually, they were either untreated or treated by metabolites coming from conventionalized mice, metabolite coming from the germ-free mice. We treated the mice with deferoxamine or DFO here because that's an iron chelator. The iron chelator will initiate the response of HIF2. I can see over here there's a robust expression of HIF2. And then if you treat this with these normal microbiota, because they're conventionalized, you can see that HIF2 expression is downregulated, but this expression is completely gone here when the metabolite is coming through the germ-free. So it says that you can actually see, if you look at closely, you can actually see the response. But we had to treat the cells with this iron chelator to induce the HIF2 and see what happens. So that I still remember this experiment. It was a weekend, and I had to go to the lab in the morning. And then when I saw this data, I knew that it had not only made my day, I knew that it had made the project. So and this is the very first experiment. It came out so well. The Western was looking good, and that directly goes to the paper. So the now the question is, gut microbiota, gut microbial metabolite is basally causing a HIF2 downregulation. But going into the detailed mechanism of that, we also wanted to look at the microbiota, how the microbial aspect of the dietary iron. So what happens when we have different irons? What happens to gut microbiota when you have different irons in the diet? So to do that, we did a microbiota analysis in the SPF mice now. So I'm just explaining this microbiota analysis because the analyzing the microbiota, we actually learned the hard way. We had some good collaborators; they are microbial experts. So we actually did our experiments in different cages, put them on these diets, and we saw that the data doesn't make any sense. then we went to these people, and they told that no, we have to co-house them, because there can be a lot of cage variations. If you put the mice on different cages, you have to actually normalize the microbiota by co-housing them with a basal iron, or same amount of iron. So what we did actually, co-housing means we actually, uh, because you cannot co-house the adult mice if they're especially, different genders, or male, they will fight. So what we did is that we actually changed the bedding, or moved the bedding, so that they have their microbiota through the feces and everything, they get normalized, because mice is actually coprophagic, they eat their feces. So that's how we kept them for 2 weeks. And then after 2 weeks, we put them for another 2 weeks to further normalize their diet, within the cages. Then we put these mice, shifted this to some of the mice to iron deficient diet for another 2 weeks. We know that this 2 weeks is good enough to make them anemic, which I just showed in this, in the previous slides. So, and then we looked at this microbiota, at their duodenal content and feces, again the upper intestine and the lower intestine, for microbiota analysis. So what we look at it is that this is the microbial analysis of these, 350 ppm and low iron diet from both the upper intestine and lower intestine, except for one, this group, which is lactobacillus. You see, at least in our hand, we do not see much of a difference over here. Some difference you see, that's not statistically significant, or I should say that, no other group was looking as significant as these. So we looked more closely at the, OTU values of this duodenal content and the feces. We see the lactobacillus is the one which is significantly up. The question is which lactobacillus. There are several lactobacillus. We found that out of, 8 or 10 species of lactobacillus that our microbiota analysis detected, we found this L. jolsoni and L. reuteri is the one, actually, those are the ones that was significantly up in both the upper intestine and the lower intestinal contents. So overall, now we see that dietary iron modulates a few bacterial population. In our hand, we saw the lactobacillus is the one which was robustly upregulated. The question is what it is doing. Then we actually wanted to look at one of the components of lactobacillus reuteri, which is known as reuterin. So we focused on the reuterin because one of our collaborators suggested that we should look at reuterin, because reuterin is a very well-known antioxidant, a component of lactobacillus. So we first measure the reuterin concentration in the intestine. And as you can see, of course, in the colon, reuterin concentration is very high. But these are the physiological means that we see in our hand, in the mouse intestine. When you looked at the reuterin level in the low iron diet, as all the sections of the intestine, small and large, you see that this is robustly up. So this actually corroborates the finding that in iron deficiency, lactobacillus reuteri is upregulated, and then you have very high amount of reuterin when you have low iron. The question is what this reuterin is, or is this return is doing anything to our mechanism that we see, that the HIF2 suppression in our system. So before that, I would actually combine our microbial metabolite component and this microbiota analysis component into one slide. So we actually also wanted to look at the uh, what are the other microbial metabolites that we can find from these, as a HIF2 inhibitors. We actually did a screening assay from this paper. We are fortunate enough to find this paper that they have, a good list of microbial metabolites that are, in order of abundance. We took 100 most top 100 microbial metabolites. We thought that abundance will be a factor to determining this HIF2 response. So we actually did a very simple experiment, took these 100 most metabolites and did a DMT1 promoter luciferase assay. As you know now, that DMT1 is a HIF2 target, so it will be read out. So we took like 96 well plate and plated, and then uh, transfected with this promoter luciferase construct and treated with this metabolite, and see which ones are effectively downregulated these things. So we got a good amount of least 8 to 10 metabolites that does that. And we did bunch of other assays, mouse experiments, in cell line experiments, and we found that only 1,3-diaminopropane at the physiological level significantly suppresses the HIF2 response. And now this Returin was not in this list, but Returin also physiologically suppress this HIF2 promoter luciferase activity. Sorry, DMT1 promoter luciferase activity, and so which is a readout of HIF2 alpha activity. So if you look at the Western of again the HCT 116, we had to treat the cells with DFO again to see an induction of HIF2 response. Now you see, and this is the physiological range of this 1,3-diaminopropane or DAP. You see, it is suppressing HIF2 in a dose dependent manner in here and in here, and at higher doses it is completely gone. And just to tell you, these doses are non-toxic. So in short, there are a lot of experiments in here, but in short, we also have proven that these chemicals, diminopropane and Returin, downregulate HIF2 alpha activity in vivo. So this is in vitro. In mice also have shown that they downregulate HIF2 alpha activity in vivo. And how do they do it? They actually block the interaction of HIF2 alpha with its partner ARNT, which I have shown in the previous slides, that it needs to be partnered with ARNT or ARNT to do this gene transcription of the target genes. So it leads to inhibits this HIF2 alpha and ARNT interaction and decrease the HIF2 alpha stability, basically, and that's why you see the degradation. And we also, there are another very popular isoform for HIF1, which discovery of HIFs was awarded with Nobel Prize in Physiology and Medicine a couple of years back. So we also had to look at this HIF2 alpha, HIF1 alpha expression and activity. So they are specific to HIF2 and they do nothing to this HIF1 alpha expression. Now the question is, can we use this finding to treat iron disorders? So if we are giving this microbial product, let's say diminopropane, and returning to iron overload patients, will it lead to normal body iron? On the other hand, if a reciprocal question is, very valid question is, if microbiota is taking iron from our diet, can we just block the microbiota or limit the microbial population in the intestine and help the host to have more iron by treating them with antibiotic? So in anemic patients, can we do that? So we actually did an in vivo experiment in here, and this is the model that we used. HAMP is the another name of hepcidin. So if you remember, hepcidin is the one is needed for proper iron regulation. So if you don't have hepcidin in the system, it will actually keep on taking iron, and the mice will be iron overloaded. So we use this hepcidin knockout model, but to do that, we have in albumin Cree, which is a tamoxifen inducible model. So we injected the mice with tamoxifen, uh, and that will eventually lead to iron overload, severe iron overload in a matter of like 2 weeks. So what we did, we started treating the mice with DAP, diminopropane, or directly given the bacteria, L-reuteri. L-reuteri is a probiotic. We given the probiotic, and the probiotic goes in and the produces reuteri. We confirmed that the intestine produces reuteri. I'm not showing the data over here, but what it does is that if you look at all the transporters that we're talking about so far, DMT, ferric reductase or T-side B, and ferroportin, all of them are hugely upregulated when hepcidin is knocked out. And as I told, that hepcidin knockout would create a state of intestinal hyperabsorption. But on the top of this hepcidin knockout system, if you're giving the mice this drug individually, this expression is completely abolished. So what it does to the systemic iron level, you can see here. If you look at the liver iron level, pancreas and heart, you see the controls. For example, in the knockouts, there are huge amount of iron is getting into the system. They're depositing. So this is a good model. Hepcidin knockout is creating systemic iron overload. Now we can see if you are with these chemicals or drugs, diminopropane and deuterin, the systemic iron level is significantly improving. So we can confidently say that gut microbiome metabolites probably can be a good target to prevent iron overload in a disease model. So on the other hand, as I just told, what happens if you are using a gut specific antibiotic? You don't want to just use any antibiotic, for example, which has many other systemic effect. Rifaximin is used for traveler's diarrhea, but it is have very minimal systemic effect because it is remains in the gut. It doesn't get absorbed, or very minimal absorption. So we use this uh, antibiotic to treat the mice. And we actually see over here, then you see the, now look at the transporter response. I'm showing the another transporter here as well. If you look at here, in the low iron diet, the transporter is upregulated. This is also upregulated, just you don't see the because of the the magnitude of the induction. If you give the antibiotic, you see you can potentiate to around 200 fold, even more in the this transporter. That's why I wanted to show this transporter here as well. So which means you increase the transporter, and you know cure or revert some of the components. I'll explain briefly why you don't see a major change in hemoglobin, because we all only did this experiment for like 10 days, and in 10 days we see that the RBC hemoglobin and RBC hemoglobin concentration, which are a good indicator of recovery in the field, it is connected. So overall, uh, we can say that depletion of gut
Nupur sir,
microbiota. Yes,
sorry to interrupt you,
sure,
but I have one question, or better,
please.
I have one query regarding this. Now we
Yeah.
are now in this slide. You are showing that you have used this antibiotic, which is specific for the gut specific microbiota.
Mhm.
Earlier we have the slides where you have shown that two types of specific mice were used. One was completely deployed of gut microbiota, and one was specific pathogen
Pathogen
deprived
free.
mice. Yes, free
Yes,
mice were there. So how
yes.
this complete the second mice, which was depleting all the microbiota, why now we are you why it is required to use or treat the mice with this kind of antibiotic which will be depleting? Why could not we use the earlier mice in these experiments?
A very good question. So we wanted to, we were thinking from the human perspective. So yes, we could use the earlier mice. We could use another model of a microbiota depletion by using a broad spectrum cocktail, right? Is that your question?
Yes, yes. So why
Yeah.
we
So
haven't used that? Why you haven't used
exactly. Yeah. So we wanted to use, this is a much milder model, by the way, because we are only using one antibiotic. We are not using broad spectrum. But from the human perspective, we wanted to test, for example, those microbiota we are using for a short term, 5 to 7 days at the most, to deplete the microbiota in the gut. But they have systemic effect. For example, we're using penicillin, vancomycin, streptomycin. There was a broad spectrum cocktail, and that was just as a proof of principle that by depleting microbiota you can have the effect. But here we're actually focusing more on the translational aspect, as if to have a safe drug and also a feasible drug, can we now test this to see what happens. So that's why we're using, and I'm sure if we did not have to do the experiment, because we actually show that this is this is working here, and that's why we wanted to test it. But we did some initial experiments, and then saw that your in the low iron conditions, actually you can go even potentiate further in those groups.
Okay. Okay.
So
Thank you.
that's the reason. Actually, it is mainly from the disease perspective, because we wanted to actually go translationally, see that if it is feasible for humans, basically. So this is the human drug. I hope I have answered the question.
Yeah, yeah, sure.
Yeah.
Thank you.
So I'm almost at the end of the talk here. So overall, the what I'm trying to show here is that how, this is in schematic. You can see there is a, this is the intestine. There is a, happy bacteria, and it is taking iron, which are these, from the intestine. This is esophagus, stomach, and the duodenum. And through the process, it is actually, taking processing by processing microbiota. It is delivering microbial metabolite to the system. And we have identified, two uh, microbial metabolites that can regulate DAP and return, that can actually control our systemic iron levels. But we can use them in our favor, where we can, in an iron overload model, we have shown that we can use this to rescue or protect from the systemic iron overload. So in short, bacteria steal our iron, but in turn they release chemicals with therapeutic potential. And for sure, not only just for iron metabolism, but novel probiotic compounds hold the secret to the future medicine. The problem is that it brought back a lot of other works, and I have not discussed the new aspects or the unanswered questions that we had in here. But the our lab is working on those. For example, I did not tell how gut microbiota, or whether it regulates the iron storage pathway in the cell, because iron storage is also a very important component in our cellular iron homeostasis and systemic iron homeostasis. So two PhD students are actually working on that, the role of microbiota in regulation is in cellular iron storage pathways. But I will not linger the talk much longer. Thank you so much for your attention. But before ending, I'd like to acknowledge the whole lab. These are the people from the lab. Dr. Yatrik Shah, I have mentioned, is an excellent scientist, very hardworking, even more hardworking than all of us. And I've got a very good training, and I'm now looking for independent positions as a tenure track faculty. But I would also like to emphasize that these are the labs that we received huge help. Gabriel Nune is a big name in the field. In the inflammasome, they have a lot of microbiota and inflammatory models. Dr. Jason Spence, who actually works on this, I have not shown the data in here, but they also work on this intestinal organoid. What they do is that they have in the literally produce intestine from stems, intestinal cells from stem cells in the dish, and they look the behavior. And these are the other people, and including our animal lab facility and germ-free facility. Germ-free mouse facility is one of the topmost mouse facility in the US, actually. So it is quite expensive, and to maintain the mice over here, to maintain a completely microbiota free facility. But could not have gone anywhere with the help of the microbiome explorer program in the university. Is a fantastic. We are still very inexperienced in the microbiota. I'm not a microbiologist, nor my mentor was. So but we learned as we are going from our mistakes, and through some diligence, we thought that we did something. So this paper was eventually published in Cell Metabolism 2020. This is an old story, but I thought this is an exciting journey. So that's why I wanted to share this. And thank you all so much for your attention. I will just end this slideshow over here.